Hello students, Mr. Azali here, and what you see on the whiteboard is my brainstorm for all the different parts, body systems, tissue, organs, and then in red is cells, different types of cells that we might be able to observe in a squid dissection. As you can see here, these are the squid. Lolio opalescens is the genus and species name, and if by chance you want to try this at home, I would encourage you to check out your local grocery store or ask your parents if they can put in an order. Maybe call ahead and see if they have frozen squid. Usually they just come in really large packages. So if you want to fry them up and make calamari, it's a food grade kind of thing, so they're safe to work with. And yeah, it's not really a problem. So I thought we could just dive into the squid dissection here after much anticipation. So you should be generally familiar with the external anatomy of the squid, and we can go over that first. And so let's see here. Let's find one specimen that we really want to focus on, and I'll keep the other one sort of put away for now. All right. Put a lid on those guys. Doing some work outside today. I want birds to swoop down and get all excited about a free meal. All right, so get this all set up. On the exterior of the squid, you can see up here I've got the fins, and the fins help steer and change direction when the squid is swimming. This is the mantle, the large part of the body. You can see the eyes on either side of the head. Massive eyes for the size of the body. And their eyes are a lot like ours, but we have very separate lines of evolution, squids and humans. And so that's a really great example of convergent evolution. That's where, through natural selection, species end up with really similar parts from different processes or different ancestors. Down here in the middle of so we're going to have the arms and the tentacles down here, and down here in the middle we've got the mouth and the beak. Might be referred to as the buccal mass. And right in there, attached to that, right behind that, is the brain. So you can see how the brain is actually in its head. So let's see here. Other important features. So this is still the mantle. You can see in the mantle there's an opening. It's just wide open in here. So the way that the squid does jet propulsion is it pulls water in to its mantle and then shoots it out the siphon. And the siphon's also like a hollow tube. So these are all muscular structures. So it pulls water into the mantle and then shoots it out the siphon this way. And then it just can do jet propulsion through the water. It makes it swim super fast. It's one of the fastest swimmers in the ocean. It's the squid. Some species of squid are considered the fastest invertebrates in the world. So no backbones. And so all of the major other organs are going to be up inside of this cavity. So we'll be able to get to those here shortly. Unfortunately, in the freezing and thawing process for this squid, we lost some appendages here. So yeah, they're not doing so good. So the arms there's arms and tentacles. Arms have suction cups all throughout. So all these are suction cups, so that means these are arms. And we've got a tentacle over here that's just about severed off, and it should have a pad. We also have one that is severed off, and it has a pad at the very end that should have tentacles on it, but that's real hard to see right now. So those structures are so thin that they easily break off when you're unfreezing them. And so let's get into it. First thing I like to do for a dissection is, well, what we just did in exploring the external anatomy. And so we're not going to cut into it just yet, but we're going to kind of explore just a little bit more. And one thing that's really cool to do is just disrupt the surface of the skin here and see if we can get it to change colors. I'm not seeing a whole lot of change, but these tiny little spots, the tiny little spots on the surface of the mantle, really the whole body, those are called the chromatophores. And when we rub these, they do change colors. Yeah, now I'm getting more pink color in here. And so by changing size, these little cells, or sometimes they're referred to as mini organs, actually, will change color. And that's a major way that the squid can defend themselves, by blending into their environment, but also by, that's how they communicate with one another. So you got these big old eyes, and they can actually talk to each other by changing color. We don't quite know their language, but you know, the closer you look, the more you realize there's a lot that we humans don't know at all. We've got some pretty good ideas about a lot of things, though. So, another thing I'm going to point out is that there's this rigid structure here called the pen, and we're going to pull that pen out in just a second. To start my dissection, I'm going to flip the squid over so that the siphon is on top. So here's my siphon. Hello, siphon. And here's the mantle. And the first cut that I make is going to be right up along this section here. So I can open this up and take a look at the different internal structures. I like to splay them out because they look like kind of cool that way. It's really simple to cut through. It's not a very rigid structure at all. And by cutting through, right away, I can recognize that in this area at the top is going to be the sperm. And so we know right now that we have a male squid. A female squid would have a much more yellowy structure in here. And that's what would turn into eggs over time, or in the right time. There's some feathery structures here that I like to take a look at that are the gills. And the gills are pretty cool. And what we can do is just cut these away and plop them into water. I'll just do one for now. And I plop it into water because it'll fluff up over time. I tried to use a little bit of warm water to help expedite this process, because we all know that if the molecules are a little bit warmer, they'll move a little bit faster. So you can see how that fluffs up nicely. Hopefully you can see. And that's where you know it's the gills, because they have to maximize their surface area to extract oxygen out of the water so that the squid can breathe. That's what gills do. Kind of similar to our lungs, but they extract oxygen out of the air and transport it throughout our body and our blood. And so these guys have blood too, but it is clear, so it's very hard to see. Hello students, here we go for part 2, squid dissection. We are into the internal anatomy, and what I've done is gone and found of my specimens a female. If you remember from part 1, the male we can tell that squid is a male because of a milky white structure up here, which is going to be the sperm. This squid has a lot of extra stuff in it that I don't remember seeing in other dissections before. So there's all these white little stringy things. I don't know if that was the contents of one of the organs or potentially some kind of parasite. I really don't know. I hate to speculate, but it's very interesting and pretty strange. So Might be something to look at under a microscope to see if those are actual separate worms. Maybe I'll put them a few into the water and see what they look like in there. So, when we look at this specimen over here, we've got a very clear identification as a female because there's these sort of yellowy, somewhat a little bit transparent, a little bit gel-like looking mass that would become the eggs. And you can see that the male doesn't have this large gland here. This is called the nidamental gland, and that's very important for female reproduction. In our female, which is totally intact, we've got both of our gills hanging off to the sides here, and connected to each gill is one of the gill hearts. Squid have 3 hearts. The third heart is meant to be right here in the middle, and it's quite difficult to actually see clearly, and that would be the systemic heart. So they'd have one part of the heart, or one heart connected to one gill, another heart connected to another gill, and then a central heart to pump the blood out to the rest of the body. Right here, this black, white, silvery kind of structure is the ink sac. And the male actually has, looks a little bit different. It's always good to look at 2 different specimens next to each other because you can kind of see the differences between individuals, just like how you and I look a bit different. And if I take this ink sac and I can puncture it, I might be able. This is a really small one. Sometimes they're really big. So maybe I can use it to write something like ink and whatever else kind of design you might want to make. Yeah, I run out of ink over here. I wrote ink with the ink sac. All right, moving forward. If we go back to the location of the mouth and the beak, you can actually get in here. If I hold it up like this, I'm going to squeeze it and I'm going to pull this beak out. And this beak is sharp and pointy, just like a bird's beak. It's pretty interesting. There we go. Let's see if you can see that. There's 2 parts to it, an upper part and a lower part, just like a beak. And this part's kind of coming apart here. It's hard to see. Okay. And what we're looking at right here is what was connected to the beak was the mouth. And then if we go in a little deeper, we pull this out. This doesn't always work perfectly right. But if I pull, not too hard, you can see that long string. That is one of the longest axons in the animal kingdom. And an axon is the long tail of a nerve cell. So this is this huge network of nerve running from the brain. This is the squid's brain. And this long axon is this incredible part of the nerve that helps really send messages quickly throughout the squid's body. This is a really important adaptation of the squid. This allows them to communicate or just send messages across their body really, really rapidly. And it's really kind of cool that they have that. If you're interested in the brain and how it works, axons, dendrites, and the interconnections between those different cells and those communications and electrochemical signals is really how brains work, humans and in squid. And that is a squid brain, and that is this long nerve cell or axon that's connected to it. All right. Got the brain, got the beak. Let's take a look and see if we can dig into the eye. This is going to be a lot of fluid, and this is really interesting. By that I mean when students do this in class, they find it to be very gross. But if we cut into the eye here, The eye is a lot like the human eye, and that means, whoa, it squirted out. Had a lot of students get in trouble for doing that in the past. This one was really easy to find. It kind of popped right out. And what I have here on the tip of, it's actually sticking upside down to the tip of my tweezers, is the lens of the eye, just like the lens of our eye. And this is a hard, seemingly feels like plastic structure. It's not plastic, it's made out of cells, but it's pretty cool. And when you dig into the eye, you can find this little bead, which is actually the lens of the eye. The squid stuff is pretty sticky. All right, now we have our lens right here. So another structure, maybe something that's probably the most satisfying thing to do in this whole dissection, is the pen. And squid evolved from earlier squid and other mollusks that had shells. They lost their shells over time. And by lose their shells, squid didn't actually lose them entirely. They internalized their shells to become more fluid swimmers in the water. We'll get the squid juice off of this part. All right, the eye juice. And so the pen is this rigid structure here. I just cut part of it. I just snipped. If I flip my squid over and I just snip right here, you can see that's where the pen is. And I should be able to just pull it out of its body. This part's pretty cool. Ready? Pull it out pretty carefully so it doesn't get destroyed. This one's a tough one. Sometimes you just gotta do it with your hands. Oh, this one's surprisingly difficult. It's really well attached at the top, and that might be because these squid have been frozen a couple times and thawed out a couple times. All right, here is our pen. And so, just to go over a couple of the different points that we have here, I have a beak, a lens. I'll turn this so you can see it in just a sec. This is the brain. This is that long axon, or nerve cell. All right, so that's more part of the beak here. And, of course, we have some of the ink and the ink sac. And then this will be the pen. And so we have all the different parts that we have pulled out of our squid. And so here's our little collection of different items that we have. The pen's really long. The beak is very small. The lens is tiny. The brain, this mass here, this is the long axon all the way down to here. That's actually a great specimen. I'd love to get that under a microscope. And just to review, some other parts of the squid were the gills floating around in this water right here, all fluffed up to increase surface area. And you can tell the difference between a male, the milky white gonad, and the female. It actually looks like eggs if we dive into this a little bit here. And the hearts that were connected to the gills, and then of course the siphon for jet propulsion. The tentacles and the arms should be 8 arms and 2 tentacles. The mass that we're looking inside of right now is called the mantle. These are the fins, and that is our squid dissection. I'm looking forward to your comments and your questions and connecting with you guys about this incredible organism. Hope you enjoy some of the other videos that I post. Hello students, Mr. Orzala here with part 3 of our squid videos, and I have here a substantial fossil, and I really like this aspect of studying squid. We've talked about cells, and we've talked about all the different parts of the squid and the organs and some of the different tissues. And now we get to kind of connect it to our learning from earlier in the school year when we studied rocks and fossils. This is a fossil of a squid that lived hundreds of millions of years ago called Orthoceras. You can see the patterns that kind of look similar to the patterns of the pen in the squid. But these squid made fossils so successfully, and there's lots of these around, because rigid structures make better fossils. So these squid from millions of years ago had shells. The squid that we just dissected had a pen, which we were able to pull out of their mantle that provides some structure, but is certainly no external shell. It's an internal shell, and it's what's left over after millions of years of adaptations to live in the open ocean and become more agile, faster swimmers, better hiders, and to be able to move around and get places faster. So squid changed from a form that had massive shells like this to being what they are today and having an internal shell. And that's one of the ways that they were able to survive and have been around for so ridiculously long. Please take care to record the notes, ask good questions, share some observations, and be ready for later in the week when you can celebrate your knowledge about squid. I'll see you in the Google Classroom.